Hi everyone. I'm Tony Brooks. I'm lead application specialist for genomic NGS at UCL Genomics. We're based at the Institute of Child Health in sunny Bloomsbury in the brand new Zayed Centre. So just a bit of background, we're a kind of collaborative core cost recovery core facility, and we want to ensure that all our kind of researchers have access to the kind of expertise and world-leading equipment to enable their genomic research. So we're quite a modular core facility, so we do everything from extraction all the way to analysis, and you can pick and choose what you want. So if you're very experienced and you just need library sequence, you can come to us, we'll sequence them. If you have data and need to analyze, we have bioinformatics core as well that can help with that as well. So we've been in existence for about 25 years now, and through that period we've basically expanded, acquiring different bits of equipment. I currently started out with kind of just 2 people at the facility back when it was formed, and now we're up to kind of almost 30 people. And literally as of this month, we've just kind of had the NovaSeq X Plus installed, plus the EVT system from Element, so they're kind of brand new in our lab as well. So we kind of have the full range of kind of Illumina kind of equipment in our lab that you would have, anything from the MyoSeq all the way up to the NovaSeq X now. So a real kind of ability to scale your project through us. So as a core facility, we obviously get a huge variety of sample types from people who work, from our collaborators. So that's not only human samples, it may be pathogen samples, it could be zebrafish, bee, you name it, and all different types of quality and quantity as well. So it's really important, just like Martin mentioned, to have a single prep kit that will work across the board. We don't have to keep buying in different bits and pieces, different library prep kits for different projects, because we can standardize our workflows, batch stuff together, and it makes it cheaper for everyone to process projects if we can keep things standardized. So as Jen's kind of gone through the main core kind of library preps that Watchmaker offer, obviously you've seen data from Watchmaker, but this is to show that actually when it comes into a core lab, that these kits actually do what they claim to do. So there are kind of 2 flavors of kits. There's the RNA kits, the DNA kits, and then 2 flavors of each of those. But I'm also going to talk about the kind of library normalization as well that we've been lucky to kind of beta test in our labs. So the RNA prep kits were the first ones we started working with, primarily the prior depletion kit. But there's also an RNA kit, which we now also have working in our lab and automated and working in high throughput. And these kits really, they manage to combine and reduce incubation steps, reduce number of sporadic cleanups, and this greatly speeds up the assay into kind of under 5 hours on the bench, or on the robots. We generally it's about 6.5 hours, slightly longer. And the kind of removal of the sporadic cleanups is really important because it can cause sample loss. They're tricky to automate and use quite a lot of tips. So the fewer of those we can do, the better. And the key point here is that the PCR mix, the Equinox PCR mix that's used in these kits, they're also robust to the presence of spry beads, where most of the other PCR mixes are not, which means that actually we can perform the PCR on the bead, and that means that we're doing fewer transfer steps through the library prep. And the RNA prep can be done in just 2 tubes. You have 1 tube for your enrichment or your depletion, and the entire rest of the workflow, you just keep it into the same tube. So your fragmentation, your cDNA synthesis, your ligation, and your PCR all on a single tube, and you just take off your library at the end. The Polaris kit also has the option of doing an additional step for cross-linking removal. Internally, we haven't seen any benefit for that. I think this is very sample dependent, but certainly it doesn't kind of negatively impact the sample if you do it. And so you've seen the slide as well already. I'm just going to kind of skip over this one, but it's a large range of input from 1 nanogram to 1 microgram, and typically anything above Win2 or a DV of DV200, which is the percentage of RNA transcripts 200 nucleotides or greater, seems to work perfectly well. We're quite happy that will generate a nice library with this kit. So for our first test, we used our kind of internal control. This is the commercial cell line, and we know this is kind of really, really, really perfect RNA. This is like Rin 9.8 to 10 RNA. It's completely free of any kind of contaminants. We know this because someone left it on the bench for a week and we QC'd it and it was still RIN10. And we've got loads of material, so you can go in 500 nanograms without a problem. And so this is kind of like the perfect RNA. So if the assay doesn't work with this, it's not going to work with anything really. And we have kind of 3 biological replicates of it treated with a CHK1 kinase inhibitor, and it gives us a well-characterized set of differentially expressed genes that we know, and we can use it as a bake-off for all the RNA-seq preps that we kind of test in our facility. So straight off the bat, we've got really nice libraries first go, and you can see we've got kind of quite a high yield from these libraries. The yield doesn't tell you everything though, so if you've got high amounts of ribosomal contamination, you'll get a high yield library, but most of that will be uninformative. So the only way to really know is to actually sequence your libraries. So we did that on an XSeq, and we then kind of normalized all our data sets down to 20 million reads, just so we're comparing everything is consistent. And then we have a custom pipeline which we've kind of built to kind of QC RNA library preps. Look at key kind of metrics that you might be interested in. So the first one to see is to look at see how well it compares kind of with the with RNA contamination, ribosomal RNA contamination, and what you can see is the Polaris one there on the far right. We have reduced to like really low amounts of ribosomal RNA contamination. In some cases, kind of lower than even kind of the poly-A assays, which is like really good performance. Normally, you would expect slightly higher ribosomal RNA from a depletion assay. We did see some adaptive contamination, but I'm going to touch on that a little bit later. So again, we want to look at duplication rates. So again, we're getting kind of quite low duplication rates. With Illumina sequencing, there is always a background duplication that's caused by the sequencing itself that you can't get rid of. So generally, anything kind of around 10% is kind of normal from a high quality library. I would say the two in the middle here, we've got the Watchmaker mRNA and the new NEB RNA Express assay. We think this is caused just by an underload of the sequencer, which always gives you spuriously high kind of duplication rates. I would also say those two assays in the middle were performed by an MSc student that worked in our lab, and he hadn't picked up a pipette before. Literally, he came into the lab and he was able to make libraries with the Watchmaker kit straight off the bat. So that's how easy it is to kind of use this kit. And again, we're getting like a really high alignment, which you could expect. You've got nice RNA right across the board, and high amounts of messenger RNA kind of detected as well. So once you've got a library working kind of manually, we want to see whether we can automate it. We do kind of quite a high amount of library prep, so we need kind of the automation to work. So we kind of went and kind of worked up a script on our Hamilton star. So this is this is separate from the script that Watchmaker have. This is our own internal script because we were kind of ahead of the curve for kind of automating this assay. And so we have a system now that will do up to 96 preps, and like I say, it works in about 6.5 hours, so it's super fast. It has minimal hands-on time. We also want the minimal mid-run reload, so you don't want to have to keep going back to the robot and reloading tips and reloading reagents. So the thing you reload is the beads when you need them to make sure they're homogenized for the cleanups, and the index plate, which is kind of pre-aliquoted out, so we don't get any evaporation from the indexes as it sat on the deck while the cDNA synthesis is going on. We have multiple safe stopping points, but also key, we want the data wants to be comparable to a manual prep on the bench. So if the data is not as good as a manual prep with the automation, then you're kind of not really doing yourself any favors. And also, because it's automated, we also have the sample index tracking kind of built into the system. And so these are the same control RNA samples, and these are some of the key metrics here. And you can see the top 6 here are in the Hamilton, and the bottom 3 were the same samples performed manually on the bench. And what you can see is that the automation is actually much more consistent than someone doing it on the bench. So we're getting super low ribosomal RNA, even better than we got from the manual preps. We're getting slightly higher messenger RNA transcripts identified. The only thing here is that we've got a much shorter insert size, and also kind of higher amounts of adaptive contamination with that automated prep. But if we kind of drill down into that, this is kind of the insert sizes based on the mapping locations of the paired reads, and we can see that with the Hamilton prep, we're actually getting a more fragmentation, we're a bit over-fragmented, and this leads to kind of an adaptive read-through contamination when you sequence through the insert sequence into the adapter on the other side. And what we kind of supposed was happening here was that the thermocycler on the Hamilton is actually quite slow to ramp up and down. And because the fragmentation is affected by heat and time, what's happening is because this fragmentation step is just longer as it heats up and cools down, the samples are over-fragmenting. So what we've done is we've basically, empirically, we did an experiment where we reduced the fragmentation time, and if we reduce it by about 10%, we get much more similar to kind of manual libraries, and I'll show you some data a bit later on. And just to show you that the data is highly reproducible kind of from the automation and the manual preps, everything is kind of clustering together, and we're getting kind of a very high number of kind of correlated genes between the manual preps and the Hamilton preps. And so obviously that was a control sample. What's it like in real life? So again, here we go, 96 samples we did, and 6.5 hours, about 1 hour hands-on time, really kind of high-quality libraries, consistent, all 96 passed beautifully, really high-yield libraries. And again, really consistent sequencing here again. I would say that we've got higher adaptive contamination, but we sequence these with a longer read length. And you can see the insert size here now is shot up now that we've kind of optimized that kind of fragmentation time. And because these were whole blood samples, we can actually look at globin depletion, and you can see you've got kind of really good globin depletion as well. So you've managed to remove a lot of the globin transcripts down to kind of really low kind of numbers. This is copies per million. So if you weren't depleting whole blood, normally you'd expect kind of 50 to 80% of your reads could map to globin. So we've kind of reduced and removed all the globin as well as the ribosomal RNA. So just going on to kind of the DNA prep, so 2 flavors of DNA kit. There's the one without the fragmentation, which is just end repair and A-tailing. We call this the ERAT kit. And then we have the frag kit, which has the enzymatic fragmentation. And this is kind of the backbone of the facility. You can use this kit not only for kind of DNA sequencing, but all kind of things that would require a kind of prep. So as well as kind of the PCR-free genome sequencing, converting, if you've got kind of template switch cDNA amplified, you can convert that into a library for RNA-seq. We use it for shotgun metagenomics. We also use it quite a lot for kind of target enrichment assays. So rather than using a library prep kit by the target enrichment provider, we will use the Watchmaker kit as our input to create a library, and then that will then go into target enrichment, either via Twist or for IDT. We've had people come to us with amplicons they wanted sequenced, so we've ligated on and sequenced those with the kits as well. And we also do quite a lot of low-pass whole genome sequencing for cell karyotyping. And also, we haven't done any chip, but I'll show you some data about Cut&Run that we've kind of worked on as well in a bit. So this is an image from the Watchmaker website, one of their brochures, and it was really about kind of adapter concentration and cfDNA prep. So we had some cfDNA preps to do, and so we decided to use the Watchmaker kit for this. One of the things that we were really skeptical about was this claim that you could go in with kind of 15 micromolar adapter when you have kind of really low amounts of material. So if you ever make a library, you would know that the ratio of adapter to DNA is critical to your library prep. If you go in with too much adapter, you promote the formation of primer dimers, adapter dimers, which you then have a pain to try and remove from your libraries before you sequence them, because you don't want to have too much adapter dimer because you're wasting your sequencing the Illumina adapter. So we were kind of a bit skeptical here, because normally, for the preps we've done before with both the Kappa and the NEB kit, we have actually diluted the adapter, 25-fold, 50-fold when we have low amounts of material. But we thought we'd give it a go, and it works, which was really surprising. So we've got some really, really nice, clean libraries, no adapter dimer. And again, this is from automation as well, as this prep is automated. And you can see we've got really consistent libraries at the bottom there from these preps. So we're really, really happy with this kit for cfDNA. Really high yield. And this went into target enrichment, a custom panel, and we were really, really happy with the results at the end. So one of the things we also want to look at was Cut&Run. So we get the occasional Cut&Run project. So just to kind of summarize here, the Cut&Run basically use an antibody to a protein binding, DNA binding protein, like a transcription factor. and then you have this MNase which cuts out the DNA where the protein has bound, and you can kind of recover those bits of DNA, and then you can sequence them to work out where your transcription factor is bound. So typically these types of projects, we have very low DNA input, 1 nanogram or less of material. The fragments are really short, they're kind of like 50 base pairs or shorter, and they can also be quite AT rich, which causes issues as well. And you can get unwanted presence of high molecular weight DNA as well, which you don't want in your material. So most of our kind of collaborators use kits from cell signaling technology for their kind of cut and run generation. And cell signaling also have a library prep kit, which is basically a rebranded kit from New England Biolabs. But there's a number of optimizations that have been done on this kit to kind of make it work with cut and run DNA. So I'll just kind of list them here. So there's a reduction in the A-tailing temperature. Normally A-tailing is performed at about 65 degrees centigrade. They've dropped this down to 50 to prevent the deterioration of the AT-rich sequences. It means it's less efficient, so they increase the incubation time from 30 minutes to 60 minutes for that. You have this issue where they dilute the adapter down quite significantly, so like a 1 in 150, using really low amounts of adapter. Use a high bead ratio to try and retain those really small fragments. And critically, you do a two-step PCR with a combined anneal and extension, and you do a really short anneal extension of about 12 to 15 seconds to prevent any amplification of large DNA fragments that may exist in your sample. So we took all these improvements, all these optimizations, and we applied that to the Watchmaker kit. And we had this one project that came to us, and it's a P63 transcription factor, and it's a key regulator in epithelial cells. And the key here is they had really low cell numbers. To try and get these cells is quite difficult. So the actual DNA we got going into the company, we couldn't detect any DNA at the start, so nothing appeared on any kind of tape traces we did. We couldn't register anything on the qubit, even with kind of really high amounts of input volume. And they wanted to test 3 different P63 antibodies that hadn't yet been validated for cut and run. They were commercially available. They weren't validated for cut and run, so they wanted to see which one would perform best. And also, we wanted to see whether the Watchmaker kit would outperform the kit that we were currently using, the NEB kit, and whether we could implement that going forward for our cut and run libraries. So we took one of the samples, we split it in 2, and we prepared one with the NEB kit and one with the Watchmaker kit. So it's probably quite difficult to see, but this is the NEB prep at the top. So we've got kind of virtually nothing here. If you kind of look at the Watchmaker kit, same input, same PCR conditions, there is actually a library there, believe it or not. There is it's very, very small, but bear in mind we split the sample in half, so we had half of nothing for this prep, and we still managed to generate a library. And when we used kind of the full sample, we went back and actually got proper samples and used them. We actually generated quite a nice library. There is some adapter dimer formation, so it's possible that for these particular samples we needed to dilute the adapter much further, but we were able to kind of remove that with additional cleanup and sequence these samples. And just to show you that actually they were really pleased with this kind of this result. So what we saw peaks and the top is the IgG negative control, and then we have our 3 P63 antibodies, and there's an H3K4 ME3 as well, which is that's basically just a histone modification that is normally associated with transcribed transcripts. Actively transcribed transcripts. So what we saw with our kind of 3 antibodies is that we saw nice peaks, reassuringly that the peaks were kind of correlated between all three antibodies, which kind of gives us kind of reassurance that this is actually kind of real. And also they appeared in known targets of P63 in skin stem cells. So I believe one of these antibodies, the P63 alpha, has been chosen as their antibody of choice, and this group are now quite happy. They're going back to collect more samples now for a bigger experiment, and we'll use the Watchmaker DNA prep kit for these cut and run libraries. And then finally, just the normalization protocol that Jen touched on as well. So we're currently working on an older version of this protocol. I believe it's being kind of currently actively worked on and improved. But basically, there are 2 different workflows. There's a singleplex and multiplex workflow, and they just differ from where you pull the sample. So for the multiplex workflow, you can pull kind of relatively early on. It means you're working with 1 tube, and it's much simpler to kind of process. The downside to kind of that is that obviously you can't really retain your libraries because you've kind of pulled them at a non-equimolar ratio at that point. And it works with just 10 microliters of library. Anything kind of over 10 nanomolar seems to work really nicely with the kind of TrueSeq type adapters. You don't need any custom primers during amplification, which means you can also go back to libraries that you've made in the past and use this kit, and it will work perfectly fine. It's really easy to use. You don't have to kind of warm buffers or keep buffers warm like we have done with other kind of library normalization protocols. And like I said, it's non-destructive. You can recover your library for the single-plex workflow, and it's completed in under an hour. I find the library is completely stable and double-stranded DNA, and it can be re-quantified by any kind of standard method you want. You don't have to use kind of digital PCR or qPCR like we would have to do for other types of normalization. So just on the single-plex workflow, so again, we've put in a variety of inputs. So you can see our pre and post here. So you've gone for like a CV of 74 of the kind of the pre-normalized library down to an 8.4%, so kind of better than kind of Watchmaker was seeing as well. But most of the time we're using the multiplex workflow. But the multiplex workflow, you only kind of get your kind of data out once you've sequenced. So we did this kind of exactly the same style as we did for the singleplex workflow. We did through the multiplex workflow, and we sequenced these on a MySeq run. And you can just see that we've got kind of a CV of kind of 6.5%, which is like really good. So if you did it, for comparison, if you did a manual prep and you've got kind of below 10, that's a really good kind of normalization. So this is kind of outperforming kind of what we do from a manual kind of normalization. And I would say that actually this was our first go, so this is still quite high for us, and we've actually kind of got below kind of 5% now, kind of relative routinely. This kind of assay is currently being worked up for normalization, and we hope to kind of have some data on that soon as well with the automated normalization. So just to summarize, the Watchmaker assay is a simple, streamlined workflow, high quality data from a variety of source material. You don't need to be particularly kind of competent in the lab to kind of generate libraries. As long as you can use Pipette, you should be fine. The Equinox's PCR mixer's ability to withstand spry beads. It means a reduced sample transfer, higher complexity library. It's easier to automate, and you can have single tube or low tube workflows. Polaris depletion is really, really good. Removes pretty much all ribosomal and globin transcripts, and they also really work really well with automation on our Hamilton. Key here is the Watchmaker can be modified to generate libraries from Cut&Run, and it gives us higher yield compared to kind of current protocols we work with, which could give us the ability to work with kind of lower cell numbers or generate more higher complexity data. And then the normalization kit, either in single or multiplex workflow, it works really nicely. You know, it's outperforming kind of manual and other kits we've used as well for normalization. So just some acknowledgements. This is just the team at UCL Genomics, sequencing team at UCL Genomics. And then at the bottom there, Tahoon actually did some of this work for the RNA library preps. And then we also have Rob and Drew and David were involved with the Cut&Run project. And then Daniel Cherry and Michelle Garrett helped us provide us with our control RNA. So they're quite helpful in providing us with RNA every time we need to control RNA to work with. And then everyone at Watchmaker who've been really helpful in kind of implementing their assays in our lab.
Awesome. Thank you.